Welcome to this Chemistry World webinar, where we'll be reacting to the news about the Nobel Prize in Chemistry 2024. I'm Natalie Starkey, Chemistry World's science media producer, and I'm here with you for the next hour. I'll be welcoming in our webinar guests very soon, and I'm happy to say that despite it only having been 48 hours since the prize announcement, we've got an amazing panel of guests ready to speak to you today and to take your questions. Just to point out that you can turn on closed captions if you find that helpful today. You should find the button in the bottom right of your screen, depending on which platform you're using. And I'd just like to point out the webinar is being recorded. So if you miss anything today or you want to go back and watch again, then do look out in your inboxes for a link to the recording in the next few days. Now, as I've said, with all Chemistry World webinars, this session is interactive, which means you can ask questions about any aspect of what's covered today. The Chemistry World team is sitting behind the scenes ready to look at your questions, and I'll be able to put some of those to our panel of guests very soon. So, who do we have joining us to celebrate the science today? Well, I'm going to run through a list of people and then we're going to welcome them in turn throughout the webinar. So we've got Philip Broadwith and Julia Robinson from Chemistry World, who are here to provide us all with a little overview of the winning science to get us started. Then I hope to be welcoming in Tom Whipple, who I think has just arrived in the webinar. He's been dashing back to his office at the Times from a train, and I'm glad to say he is here. If you've read any of Tom's work, then you'll know what a brilliant reporter he is, and so we thought it would be excellent to hear his opinion on this year's prize, particularly as he's spoken to some of the winners already, so maybe we'll get an inside scoop there. Then I'm delighted to say we'll be joined by Paul T. Kim, a PhD student in David Baker's lab, and also Benjamin Sanchez Lengerling, who not only worked at Google DeepMind for 5 years, so can give us some insight into the inner workings of the company, but he's also now a professor at the University of Toronto. And last, but definitely not least, we were really lucky to receive an email from David Baker last night, one of the winners of the 2024 Nobel Prize in Chemistry, who said he can join us for today's webinar. We're very excited about this. He's not here just yet, as it's still very early in Seattle, but we hope to see him a little bit later on, so we'll welcome him on screen when he appears. Now, we'd love to hear what you think of this year's prize announcements. Was it what you were expecting? And are you happy to see this area of science being recognised? Did you think maybe a different area of science was missed this year? Or do you have a personal connection with this year's winning science? Please use the questions box to send us in your thoughts. We love to hear from our audience, to see who's in the audience today, and I might be able to read out some of those comments later on. And of course, please also send in your questions for our speakers too. You can send those in whenever you're ready, because I'll be able to move through those as we go through today's session. Now, let's get us started with welcoming Philip and Julia from Chemistry World on screen now, if they'd like to turn on their cameras. Hi, Philip. We'll just get Julia, there she is. Hi, I would just give the audience a little brief overview about the two of you so they know what your expertise is and what we're all doing here. So Philip has a PhD in organic chemistry with 15 years journalistic experience in various roles at Chemistry World. And he's got a broad interest in all aspects of the chemical sciences and their applications. As business editor of the magazine, his focus is on highlighting the science going on in commercial settings, as well as companies' efforts to address the grand challenges facing industry, society, and the planet. And Julia Robinson joined the Chemistry World team as science correspondent in May 2023, and we're really lucky to have her. She previously spent 8 years leading the clinical and science content at the Pharmaceutical Journal, the official journal of the Royal Pharmaceutical Society, which is a membership body for pharmacists. So welcome to both of you. Let's get started about talking a little bit of an explainer about the science. I think that's probably a really good place to start. I'm going to hand over to you two to kind of give me a bit of an explainer, because I think that would be really, really handy.
Okay. Well, I think it's probably a good place to start to just talking about proteins themselves. So this probably isn't news to a lot of people listening on this call, but proteins are the chemical building blocks of life. So every protein is made of a string of various combinations of amino acids that fold up into a specific 3D shape or structure. And it's this structure that is what gives proteins their function. So, whether that's becoming muscles or hormones or enzymes, there are just so many different types of proteins that all have different roles. But the thing with proteins is this interesting 3D structure. So understanding a protein structure helps us understand how it works. And if you can figure out how proteins work, you can understand more about how they interact with other molecules, as well as design entirely new proteins with desired functions. So new drugs or vaccines or catalysts, the possibilities are huge. But actually getting to that point of being able to see the structure and understand the structure was something that consumed scientists for decades. So at first, I think what was quite difficult was having the right tools to be able to determine the structure of proteins. So go right back to the 1950s, where X-ray crystallography kind of enabled us to get the first 3D structure of proteins. This work actually also received a Nobel Prize, I think in '62. And other experimental methods like nuclear magnetic resonance, cryo-electron microscopy have since been added to allow us to determine the structure of thousands of different proteins. Then in the '70s, it was sort of discovered that it was the sequence of amino acids that determined the way that a polypeptide chain folds itself. Again, another Nobel Prize. And this led to the idea that you can, in theory, predict the shape of a protein just by knowing its amino acid sequence. But it's a neat theory, but actually a pretty difficult one to prove because there's just so many possible conformations a protein can adopt, and being able to explore them just all takes just an incredible amount of time. So there's another sort of 50 years until we kind of got close to that. And they actually kind of introduced an initiative called CASP, which was to encourage researchers to work on this, and it became a competition. Actually, the Nobel Prize winners entered this competition, and it was kind of only when AlphaFold 2 came along that it was kind of proclaimed that this protein prediction problem had been sort of largely solved for single proteins. So I don't know if you want to add anything.
Yeah, I was going to say, if you think about a protein, you think about the way it folds up, it depends on not just the sequence of amino acids, but all the interactions between them, right? So you've got some that are charged, you've got some that like to be near the water side, the outside of the protein, some that like to be on the inside, if they're hydrophobic. So even if you know the sequence, there's millions and millions of different ways that sequence could potentially fold up. And if you make even tiny changes to the sequences, you can make completely different folding patterns. So predicting that even from the sequence is a hugely difficult thing to do if you want to try and do it by doing all of those interactions. So the thing that I think AlphaFold did quite differently, and the application of AI particularly, is saying, well, rather than trying to kind of model all of those individual interactions, we'll just look at generally how proteins fold. How does the sequence end up with that three-dimensional shape? And then we can feed in new sequences and see if we can generate structures. And that's particularly important because it's really, really difficult to physically get a lot of proteins to crystallize, to be able to measure their structure, or to be able to see it in NMR. Those techniques are really, really difficult. They take huge amounts of time, and several Nobel Prizes have been won for individual protein structures. But to be able to do that for all of the thousands and thousands of proteins that are out there would just take it, and for some it's impossible. They don't even really fold up into a kind of defined shape. So the advantage of AI is we can say, well, we'll take all of the things that we know about how proteins fold and use that to make this model that can then make predictions. And the thing that
yeah, I was going to ask you what's the difference between the two prizes, because I think that's, it sounds like they're very, very similar, and they've obviously been awarded the same Nobel, but there's obviously a subtle difference between David Baker's work and that of DeepMind. So are you able to elaborate on that a little bit?
Yeah, sure, I can do that. So I think this Nobel Prize is really interesting because it kind of, as you say, it recognises sort of two different discoveries, but the work is just so closely interlinked. So Hassabis and Jumper, they developed AlphaFold 2, as we know, which predicts the 3D structure of proteins based on their amino acid sequence. And then Baker, so he developed computational software called Rosetta, which kind of helped solve like the inverse problem. So starting from a desired protein with a particular structure and then going backwards to kind of figure out what amino acid sequence it would have. And this allowed him to create entirely new proteins that had never been seen before. And it's called de novo design. And so that's kind of the difference between them. They're sort of, one's kind of going one way, one's going the other way, but they're both just sort of equally important and enormous breakthroughs for the field.
Yeah, and I think we'll be hearing more about that from one of our other guests, Paul, later on, because I think that's exactly what he's working on, in fact, that de novo part. So that's great. I've got a question about, I think since the announcement of the chemistry prize, but also the physics prize this year was heavily AI, machine learning related. There's been quite a lot of, social media presence about this, about should we be worried about AI? Is AI going to replace humans? How important are scientists in this process still?
Yeah, I mean, at the moment, AI is very much still a tool, right? It's a really, really good tool in certain things, and it can do things that humans can't do. But there's also a lot of things that the AI can't do that humans can do, and that's the kind of the thinking, the idea generation, the knowing which questions to ask the AI, right? The AI is great at saying, here's a load of data, I can find some patterns in it, I can give you an answer if you ask the right question, right? Mm-hmm. And it's the knowing which questions to ask. But you'll see, you may have seen interviews with one of the physics Nobels, Geoffrey Hinton, saying he's actually quite worried about the real long-term future aspects of AI. How do we ensure that there is a kind of safety factor built in, that the AI can't kind of take over and do those things. So I think that's a real worry, especially given that the people who invented it are slightly worried about it. But it's still quite a long way off from where we are right now, and we have the opportunity to kind of build the appropriate safeguards around it.
Yeah, and I think using it as a tool, isn't it? It's knowing that we're using AI, but I think it's like a lot of the scientists come out and say, well, we still need the scientists. We need those, we need our brains of humans to actually interpret that data and understand that we're getting something reliable and sensible. So I'm actually going to bring up a little poll now, because we had some polls ready for the audience, because I think it's a bit of fun, and I'm going to launch this one. And I want to know what your views are on the audience. You can click on the screen and to one of the answers. If your screen's small, you might need to scroll to see all of the answers. But the question is, a never-ending question, the use of artificial intelligence in chemistry is, and we'd like to know your thoughts on that. Because, yeah, I think it's been quite an eye-opener this Nobel year, hasn't it? That we've got this really new technology being recognised so heavily in 2 prizes. And a lot of us are still quite unsure about the direction it's going. So let me see, we've got... our audience are very positive today. I like this. So quite a few of you have voted, and actually an exciting opportunity is winning with about 60% of the votes. And 25% of the votes are saying just a new tool like any other. So we've got a very positive audience in today, which is really, really cool. I'm going to end that poll shortly. If you want to have a vote, please do. I'll end that now. Well, thank you for joining in with that. That's brilliant. I'm pleased. Maybe we've got a lot of scientists in the audience who know what they're doing and are enjoying using AI. I'm actually just going to ask you another question about, in terms of, you're obviously science journalists, you follow lots of different science areas. Was this year's chemistry prize a surprise to you, or did you see this coming?
I think in a way we might have seen it coming. It's interesting enough, when we were listening to the announcement of the Physics Award, one of our colleagues actually predicted that John Jumper might be one of the chemistry prize winners. But actually we always report on, say, Clarivate, who are the Web of Science provider. They always publish an annual list of citation laureates, which is basically researchers with the most frequently cited publications. So we always report on that every year as sort of a bit of a prediction for who might win the Nobel Prize. And actually on that list were all 3 of the winners this year. So there were also other people on there as well. So it wasn't a given, but I think we were all sort of, slightly expecting it, I guess. But it's always exciting to find out who, and always a little bit surprised.
It is, and I know. I mean, being in the Chemistry World office, I know it's like it's chemistry Christmas for us. It's always a very exciting day, and it's always nice to see chemistry being celebrated. And there definitely is a lot of chemistry in this prize, even though we've talked a lot about AI here. We're going to get on as well to more chemistry, and it is definitely a chemistry prize. At this point, I'd love to bring in Tom Whipple. Brilliant. Hi, Tom. Nice to see you.
Hello. Hopefully you can hear me all right. I haven't tested these.
Yeah, you're quite loud, but that's okay.
Oh.
We've got you loud and clear. So, as I said, Tom is science editor at the Times. He's rushed into the office to join us, which I'm really happy about. I'm not going to read out your whole bio, Tom, because people can have a little read of that, and they probably already know who you are. But yeah, tell us, what was your reaction to this year's prize?
I think this is the first, other than CRISPR, this is the first I've ever guessed correctly. I was pretty certain that, well, I think we all knew they were going to get it one time, and it just seemed an obvious one. But my reaction to this prize, I've been following DeepMind, as a lot of us have, since sort of the middle of the 2010s. I first interviewed Demis in 2018. The weird thing about Demis is that he sort of has this all planned, kind of from about the age of 12, when he was the second best chess player in the world, who was aged 12. And then he sort of decided, he brought together the components of what was needed for him to make an artificial intelligence company. He started with programming, and along the way he just programmed Theme Park, that multi-million selling computer game, age 15. And then he went to do computer science at Cambridge, and then he did a neuroscience PhD, and then he started DeepMind. And I spoke to him yesterday, I asked him, is this a surprise? Is this sort of pretty much where he expected to be? And yeah, this is pretty much where he expected to be, kind of solve the proteins. He said in 2018 he'd solve proteins, and then obviously get a Nobel Prize. And the point of that is he's planned the entire thing. This is where he expected to be. The next stage in the process is the artificial general intelligence. And he's not so Jeff Hinton gave that sort of oddly quixotic Nobel acceptance speech where he said that, thank you for the prize, and I do hope my invention doesn't subjugate humanity. Kind of Demis isn't as worried as that. He said he's cautiously optimistic that AI isn't going to kill us all. So that's good.
I'm glad to hear that. Yes, he's quite a character, isn't he? I mean, I looked up when he won, I was like, as everyone looked up who these people were. And, reading his Wikipedia page was it was incredible. It was almost just ridiculously unbelievable. And I was like, but I think it's all true. And as you said, you've spoken with him, and it's amazing to see that, I think I just needed to read the first few lines of his early education to go, oh, actually, he's probably going to win a Nobel Prize in his future somewhere, because it just comes across that way. And obviously, he is this incredible mind. And you've met with him, I take it, this week. You've had a chat with him? How has he reacted?
I met with him. I last saw him a week ago, actually. I was at a Times event. I spoke to him on the phone yesterday. He is the other weird. I don't want to make this entirely sort of here are the quirks of Demis, because the most tediously annoying thing about him is he's a pleasant human. I don't know if he's used his great intellect to sort of work out how to interact with normal people, so he's not annoying, but he just comes across as a normal guy. But he works. He's a second day. He works every day from midnight to 4 a.m. And that's when he does his research. His first day is when he runs the artificial intelligence branch of Google. Last night, in fact, he did let his hair down. I've never seen him at a sort of DeepMind drinks, but last night, to celebrate the Nobel, he let down his hair. He had a poker night organised with his friends. His friends are several poker world champions and Magnus Carlsen, the highest rated chess player in human history.
Right. Well, I'm glad he has fun that way. That's great. That is quite incredible. But I love that. And I'm just wondering, have you spoken to John Jumper? We've not heard so much about him because he's not quite got this, big character as well.
No, he hasn't got that sort of thing. And look, it's ridiculous if you're like the Ringo Starr of Nobel Prize winners. He's absolutely not that. He's very young to have got a Nobel. I think, and I've met him a couple of times. I went for a drink with him. He does turn up to DeepMind drinks, and he was at their summer drinks this year. He's an absolutely lovely guy. He's an American. He studied at Cambridge, and he decided to stay here. And he's headed up the protein folding division of Google DeepMind. And obviously, a lot of this is down to him. I mean, as far as as far as the Nobel Committee are concerned, he's got half the prize, and he absolutely deserves it. He doesn't have the, I guess, the sort of the intriguing mystique of Demis. I sat down and had a beer with him, and you wouldn't know he was going to be a future Nobel laureate, except that, I knew what he'd done. He's a lovely guy.
Yes, definitely. I saw their press conference and they both came across really well and explained their science really well, which is always really nice to see people that can communicate what they do. And, I also watched David Baker one. We're going to be welcoming David Baker hopefully on screen just later on in the webinar. What do we know about him? Do we have much on him? I'm excited to talk to him later. He came across great in his press conference. And I thought, wow, he's just seems like a really great scientist. And, do you think it was the right time for him to be awarded? This is quite relatively new science in a way. How are we feeling about that?
Well, I mean, he's had longer to wait than the other two. I am not a David Baker expert. I'm going to slightly recuse myself in a moderately chauvinistic fashion of the British press. It's the Brits we're interested in, and we want to. John Jumper is an honorary Brit for this purpose. Demis Hassabis is a Brit. David Baker has the great misfortune of living on the west coast of the US, and thus hasn't made it across our radars. Look, absolutely, I congratulate him, and I'm peripherally aware of his work, but I haven't had any contact with him as a human, and I'm sure he's a lovely one.
Philip, I'm going to come to you. In terms of David Baker's science, what does it mean for chemistry? I want to get back to kind of, we've spoken about the AI a lot, coming back to the real chemistry components here.
Yeah. I mean, so we've been kind of covering David's work at Chemistry World for as long as I've been at the magazine. He's kind of popped up every now and again, fairly regularly. And whenever you see a paper by him, you kind of go, well, this is worth looking at because it's always really kind of interesting and insightful, and it's really exciting. Because, I mean, we started, the early stuff that we started looking at was kind of, taking an existing protein and modifying a bit to be able to do something that a natural protein can't. So for example, to catalyze a chemical reaction that doesn't exist in nature, but we know about from chemistry, but we might want to be able to make a catalyst that can do it better than we can do in a flask. So there was that. And then this kind of whole idea of being able to make proteins from scratch to do pretty much whatever you want is hugely exciting, because if you think about the things that proteins can do, both in a biological and a kind of chemistry context, the possibilities are enormous. We can start to develop catalysts for chemical processes that run at lower temperature and are much more environmentally benign than existing catalysts. We can start to look at designer medicines and things that work a bit like antibodies but without some of the drawbacks of antibodies. You can do all sorts of amazing things with nature's building blocks of kind of harnessing that to do things that nature doesn't do, which is just, that's mind-blowing to me.
Yeah, and I think we're definitely going to hear more about this later on. Hopefully when David joins us, we'll talk about some of those applications because there are many, and the list is literally endless for what you can apply this kind of science to. So it's quite incredible. I'm really excited about the prize. So I think what I'm going to do now, guys, is if you're able to hang around, I'm not sure if you are, Tom, but if you are, I'll welcome you back at the end on the panel if we've got some time. We've got lots of questions coming in, but I'd love to welcome on screen now our next guest. He's actually Paul T. Kim. So I'm just going to say goodbye to our lovely journalists who've joined us, and thank you so much for explaining the science and coming and joining us today. So hi, Paul. I can see you on screen now. So Paul is actually a grad student in the Baker Lab, working on deep learning methods for de novo protein design. His current project focuses on design of DNA binding proteins. Before coming to the lab, he worked at the New York Structural Biology Center on machine learning methods for cryo-EM and at Bayer on small molecule drug discovery with unsupervised learned representations. I'm trying to say all these words, goodness, a lot of talking today. Welcome, Paul. It's so good for you to join us. And I know it's very early for you over in Seattle. So thank you for being here. We're really glad to have somebody join us from the lab and also to talk about your work. Now, obviously, I've just explained what you do, but I'd love you to explain it back to me now in more simple terms for me to understand exactly what your work is, what you're working on at the moment, and how that relates to the wider work in the lab and the Nobel Prize.
Yeah, sure. Thanks for having me. So the lab is always trying to expand the capabilities of protein design to new domains and new targets. And one of those targets is to be able to interact with DNA. DNA, holds our genes, and like what genes get expressed or like silenced determines how the cells in our bodies behave. And so it's very key then to make proteins that can interact with that system and, upregulate or downregulate certain genes. There's already kind of natural ways to do this, basically taking proteins from nature and trying to modify them. But we want to try to now make completely new proteins that can interact with DNA and be specific to specific sequences and make new things happen.
Thank you. I'm trying to think about what initially drew you to work on what you're doing now. Obviously, I expect you've got a background in chemistry or biochemistry or something, and you've obviously been drawn to work at the Baker Lab. What kind of made you want to do that?
Yeah, I actually saw a talk that David gave back in 2019, and I was really excited by what the lab is doing. I think there's not many places in the world that do both the computational side and the wet lab biochemistry all under the same roof. And when you do that, that tight collaboration that you can have can really accelerate the speed that you can make new discoveries. And also it makes sure that all the work that you do is grounded in like very real kind of results rather than some kind of like computational model or some kind of benchmark data set. And so I thought that was really exciting. And then when I went off to look to do a PhD, I learned that the Baker Lab had gotten really into deep learning and AI, and that was my background. And so, yeah, it was a natural fit. And yeah, I think all of us in the lab are true believers in, the potential of what you can do with protein design. We're all here because we want to make that possible. So I hope, with the Nobel on the field and on David, the spotlight can kind of, inspire us to keep going and make new discoveries and deliver on a lot of this hype.
Absolutely. And actually looking at, the Baker Lab website, it's an enormous lab. There's so many of you. And when I had a look, I was like looking at all the different projects that people are working on. It's incredible. As I was saying just now, there's so many applications of this work. Can you give us a little overview of some of, the most exciting things that some of your colleagues are working on, perhaps?
Yeah, sure. Right now, the big push in the lab is for enzyme design. And so, enzymes are proteins that can catalyze some kind of reaction. They actually do a lot of the metabolism in the cell. And more broadly, also, we use enzymes to do industrial chemistry, for example. There's a huge range of things that you can do with these sort of like nanoparticles that can, like small molecules that can do catalyze new reactions. And so that we've been making a lot of breakthroughs on that front. There's also very much more kind of like long goals of trying to redo photosynthesis and make it more efficient. That's a project that I think is really exciting. There's a bunch of people working on different types of therapeutics and immune regulation, like cancer signaling stuff. Yeah, there's tons of crazy cool projects going on, and it's a really exciting place to be.
It does sound exciting. I imagine your lab meetings are quite long, and you've got to discuss all these types of science. I don't know how that works. How is it now? I mean, obviously, you've known David for quite a while. Have you seen him since the announcement?
Yeah, definitely. He was, Wednesday was a big party atmosphere in the lab, and David came up, and we all, cheered him. Then we had a, bunch of people in the lab from our friends and colleagues in other labs that came up to visit, and it was a bit of a circus. And then, yeah, we had like a big party in the courtyard afterward. David had to talk to everyone, of course. And then yesterday, pretty much business as usual. We all came back to the lab, did our work. I'm actually mentoring a rotation student right now. And so David came by to ask how that progress on that is going. And yeah, he seemed, a little more upbeat than usual, of course. But yeah, it was a pretty normal day.
So he's a very hands-on PI of the lab, it sounds, which is incredible when there's so many people. And, I'm hoping that won't change. Obviously, what comes along with the Nobel Prize is a lot of media interest and a lot of requests to go and do work all around the world and talks and all this stuff. So, I guess that's going to be interesting for him now to balance that. He's still, relatively early on in his career. So I guess it's now he definitely sounds like that there's more he wants to do, and he's not going to be turning his back on the lab anytime soon, which is really good. I'm just having a look at some of the questions coming in from the audience. We've got loads. So thank you, everybody, for sending in questions. A great one from Ruben. Has any of David Baker's work on computational protein design directly impacted medical treatments or therapies? Do you know if there's any kind of anything going on at the moment with that?
Yeah, definitely. There's lots of drug candidates that are currently in the clinic through some of the translational investigator programs that we have at the Institute for Protein Design, and also at the various startups that have spun out of the lab over the years. We've also had a COVID vaccine that was approved for use in Korea. So yeah, there's been a couple things. Not a ton yet. I think there's a lot of hype in this field, and now it's up to us to deliver on that, because yeah, it's still kind of early days for protein design in a way, and we've accomplished a lot of things. We've kind of opened the door to being able to make proteins that are really interesting, but now it's like the hard work, the 10-20 year process of actually then turning this into drugs and therapeutics that can be on the market.
Yeah, and in the early days, people didn't believe this was possible, and so I guess it's part of David's work has been turning that misconception around, saying actually we can do this. It sounds a bit crazy, but this is something that can be done as real science and be useful, which is really amazing. I've got a question from Ben. When you started your PhD, did you think this field would win a Nobel? Did you think at the time this is a groundbreaking field, it's going to be amazing, and it's going to be rewarded in the future? I want to be working on this.
Yeah, I don't know if I really thought on those terms when I was thinking about protein design, but I think after I joined the lab, we had some really big papers come out using new generative AI techniques to propose candidate protein structures that we could then use to filter on and eventually, make in the wet lab. And so those were very impactful. And when those came out, I think there was a lot of rumor in the lab that David's probably in line for a Nobel Prize, and that it would probably be shared with the DeepMind team. But of course, we never know if that's ever actually going to happen. And I think any given year, the odds of winning the Nobel Prize are quite low. So we didn't know that it was going to happen this year. But yeah, I think that this field definitely deserves a Nobel Prize, and it's something that was the right thing to happen.
And how much work does your lab have with the AlphaFold kind of the DeepMind side of it? Do you talk? Is this something you use all the time, or is it definitely two different things?
No, we're definitely in contact. We meet at conferences. There's actually a pretty big conference for protein designers led by kind of the Baker Lab called RosettaCon, and people from the DeepMind team are always over and they're presenting their work to us. And what they do is super exciting. They're always on the forefront of the kind of protein structure prediction problem. And yeah, no, there's definitely tons of collaboration. I think some papers are even co-authored by some folks at the DeepMind team and the Baker Lab. And of course, we also use some of the data that they've generated with the AlphaFold DB to train our models. That's part of a standard process. And a lot of these protein structure work is to now use the structures that AlphaFold predicted as inputs to your next generative or predictive model.
Okay, that's good to hear. I'm going to have maybe a final question for you, actually coming from the audience again. Something about how do you ensure the accuracy and reliability of AI models in predicting protein structures? I'm guessing it's like you've got to have, as we were saying earlier, you've got to have the scientists there. It's not just about AI producing all your science, and we would give the Nobel to AI then, but it's about the scientists who are using it. So yeah, how do we ensure all of that? How much checking needs to be done, and how much input is there?
Yeah, definitely. I think so. For example, I mean, on my team, we have a couple computational folks, and then myself included, and then a whole bunch of wet lab scientists with more backgrounds in chemistry and biochemistry and biology, and they're kind of the final say on all the proteins that we make. They kind of, run the models that we give them. They look at the proteins that come out. They kind of tell us, oh, these things are good, these things are bad. Maybe you should try to tweak the models in this way to try to make them better. We do those tweaks, and then we come back to them with new models, and then they try them out. And when they finally see stuff that they like, maybe also using other AI models to filter on this like large set of proteins that they've generated. Once they see stuff that they like, they'll then go in the wet lab and try them out. And that's always kind of the final validation for our work, is that it actually holds up to the real world of biochemistry.
Oh, okay. That is so good to hear. I didn't know that's how the process would all work, but that's incredible. So yes, Abby, as you said, having those two labs, the wet lab side by side, is super important for how that work is going to progress. Look, thank you so much, Paul. I'm sure we could keep chatting for the rest of the webinar, but we do have some other guests with us, and I'd really like to speak to them as well. It's like, I love that we got so many people today. But look, thank you so much for joining us. I'm going to wish you all the best with the rest of your research. I know I've done a PhD. I know what it's like being in the middle of it. It's kind of stressful but exciting at the same time, and I know you're going to do fantastically. You seem to know your subject extremely well, so thank you so much for joining us, and you'll see David in a bit. We might welcome you back at the end of this time. We're going to have to see how it goes, because we still have lots of questions coming in, but right now I'm going to say goodbye for a little bit, and I'm going to welcome our next guest on screen, and that's Benjamin Sanchez Lengerling. So we've got his bio up on screen now. Hopefully he's still around. There we go. Hi, Ben.
Hey, how's it going?
Excellent. Thank you so much for being here. I'll just give you all a little intro. So Ben's an assistant professor at the Chemistry Department at the University of Toronto, PI of the Chemical Cognition Lab. Previously, Ben worked as a research scientist at Google DeepMind/Brain for 5 years. So this is hence why we've invited him in today to talk about the other side of the Nobel Prize. Ben's a researcher who solves chemical problems leveraging data-driven techniques. He designs, builds, and evaluates computational tools that enable molecular discoveries covering small molecules, polymers, chemical mixtures, and proteins. So yeah, thank you so much for being here. Can I, before we begin, just bring up a poll on the screen, because we're going to be talking a lot about AlphaFold, and I was interested to see in the audience who's used AlphaFold, because I don't know who our audience are. It's just a number to me. You're all coming up as a number, and actually I'm interested to see who we've got. And if you haven't used it, or if you have, tell us a bit more in the questions box about your experience. If you're looking forward to using it at some point in your career. We've got quite a lot of people saying no, they intend to use it. That's about 20% actually of the audience, and actually, do you know what, the no is 72%. So a lot of people in our audience are going to need a bit of explanation on AlphaFold, and I feel like we've got the perfect person here to do that. So I'm going to end that poll now. Thank you for taking part in that. That was really interesting to see. So Ben, can you tell us, I think let's start from the beginning, a little bit about your experience of working at DeepMind. You were a researcher there, I believe.
Yeah, my research background is in computer science and mathematics, and then I did a PhD in chemistry. And I got started in Google Brain before it was Google DeepMind, because there was a team that was starting to get built up around AI and olfaction, which is a chemical sense. And so the idea was like, let's work on molecules and AI. And later then I did a transition into a team protein function. This is a team led by Lucy Colwell. And it's a team that actually works a lot on applications of proteins and serves predictions for UniProt, which is the huge database of like protein data in the world, hosted by Nobel. So, I've been working a lot on like the intersection of AI and molecules and proteins, and our team would often interface when there's like questions about structure with the AlphaFold team, especially once we open up into Google DeepMind. We before we couldn't see each other, now we're like, oh, like we get to see, like we're all working towards the same vision of trying to solve these type of problems.
And so when you say you worked with the DeepMind people, did you have any access to Demis and John? Were you working with them directly? What kind of role were they playing within that wider structure?
Yeah, so I did work underneath them. We sometimes talked with them, and we're all in the same group organization. Demis is like, sometimes you would see him if he's in a meeting, because he would be like in a lot of meetings. We have town halls, so people actually get to if you go to these town halls, often they're in London, but now, DeepMind is really a big global research organization. You'll get to talk to if you go to any of these, you get to talk to them in person. We interface with the AlphaFold team when we had we're working on protein function, so it's when there's a question of okay, what part of this is structure related, we would come with our problems and and see, and often we would also utilize the tools that the whole team was developing.
Ah, okay, that's super interesting, and I'm guessing it's quite a large team, isn't it? So trying to bring all this together is it's quite an interesting thing. I'm just looking at another question coming in, which is great actually, from one of the audience members, having seen how this field of science works in industry and academia, because obviously you're working as an academic now. You've seen it from two sides, which is really interesting. Yeah. What are the differences? How's it do in different areas?
Yeah, I think we're in a very unique time where you have large companies willing to invest in pure research. Often they are willing to do this, one, because they really believe in science, and also because they see that there's a lot of benefits of investing in new technology. There's this technique called graph neural networks that is often used in chemistry and also for proteins. And one of the main research papers was invented at Google, and now this is used in Google Maps, right? So there's often these trickle-down effects. There's a few things that I saw different, right? One is and I think this is why it's possible to actually get these kind of breakthroughs. It's really you have a long-term commitment and you have professional people working on a problem. So when you have really this mass of expertise, the David Baker Lab has, but also in Google DeepMind, you're really able to push the needle in one direction. And I think when there's enough momentum and the problem is clear enough, you're able to do it. I guess some slight differences is I think in some ways industry is sometimes slightly better managed. People work 9 to 5, tend to do that. They have families. They're we're just going to work on this. And sometimes right now, at least there's an idea of a long-term vision. There is often research sometimes dies by a PhD student, right? You finish your PhD and then you need to retrain somebody before they can take that idea, or that idea never solves. In this case, it's very team-based. So I don't think there's one that is better or the other. It's just it's a different form of working, of trying to do science. And we publish, these teams publish all the time. But there is a less of emphasis on publishing small papers, but more on trying to really get impact. That's an abstract notion, but really looking for hard problems where in this case, Google has an edge, right? Google has big computers. They have capacity in engineering for storing data and accessing it and training models. Let's pick problems where that's useful.
And I guess in some part it's a bit of a funding issue, because we know with academia funding it comes and goes, and I guess with the Baker Lab right now it's large enough that it can keep going, and it's got enough people working on the same problem, and you say it's kind of that momentum. Whereas in industry, if somebody decides they're going to put money behind something, it can keep going, and it has that momentum to keep that research ongoing. So that's really interesting that that's happened in two different ways, and getting to a kind of similar result. I've got a question from Sophie. Thank you for your question. So it's unusual for a commercial company to win the Nobel. Do you think big tech will contribute more to research in the future versus traditional university environments? That's a good question. Thank you.
It could be. It could be. Like, I think the tides are changing right now in tech. I think you see a lot of pressure, especially like a lot of these tech companies are like seeing pressure to like really, especially with like large language models, to really like it's much more competitive. So, I think tech companies are seeing that they can't get distracted that much. That said, there's Bell Labs and IBM that also got Nobel Prizes previously. And I think there's still a few still ongoing work that might get a Nobel Prize in the future. Maybe not in biology. There's really interesting physics models for climate that are potentially very impactful. It'll take time for this impact to show. Yeah. I think overall, as long as that science is being done in open source way or being shared, I think it's a net positive, right? It's different strategies of trying to still push the needle of the sphere of what we know and can do.
Yeah, and obviously historically Nobel has not rewarded, emerging science fields. That's the point of it. It's emerging stuff that is very proven. And actually probably this year it's feeling a bit more like this is an emerging field. But I guess that's why it's been so interesting to see the Nobels coming this year for these AI areas. A great question from Nandini. Where do you think AlphaFold plans to go in the future? What will they be working on? I don't know. You can't give us any inside sight that you're not allowed to tell us, but what are your thoughts on that?
Yeah, I mean, I left DeepMind 5 months ago, so I only know a little bit of what but this field moves super fast. I think that you can definitely see where AlphaFold is going with the latest paper or preprint that they put up, which is really trying to simulate. So AlphaFold, the AlphaFold 2, the paper, that the work that got the breakthrough is they work specifically in proteins, but now the newest version actually.
Ben, I think we're just losing Ben a little bit. Yeah. We can't really hear you at the moment. So what I'm going to do, I think if David Baker's joining, he will be joining very soon. I'm going to actually welcome the rest of the panel back on screen now so that we can get through some of these questions. Stay with us, Ben, because, yeah, your Wi-Fi, I think, has just gone a bit flaky. Let me just keep an eye on my box here. So thank you so much for your questions. We will try and get Ben back. Yeah, so whoever's still in the background of my panel, if you can come back on screen now, that would be great. Hi, Philip. Hi. Yeah, brilliant. We'd see who we've got. We've got Julie, we've got Paul. Okay, right. Let me get through some of these questions. Let me see. Right, got to get to the right box here. A great question. It might actually be for Ben, but it might not be with us still. We'll see. How are the challenges of different in terms of computational protein design versus polymers and small molecules and mixtures, etc.? Paul, I don't know if you can comment on that actually. So yeah, in terms of designing these polymers and mixtures, how different is it when you're looking at the computational protein design?
Yeah, it's interesting. There are some, there are definitely trade-offs. So a protein is in some ways a polymer. It's a polymer of amino acids. With proteins, you have actually a sort of more restricted design space than you do for small molecules. Like the space of possible small molecules is vast because you can arrange this graph of atoms and bonds in very many ways, whereas a protein sequence is a 1D string with 20 different possibilities. for each position in that sequence. The space of possible proteins is also unfathomably large, but the space of all possible small molecules is like an unfathomably even larger space. The drawback of that, though, is that the space of molecules that are synthesizable is actually a much smaller subset of this. And so there's a lot of molecules that you could try to, theoretically come up with on a computer, but then there's actually no way to synthesize that small molecule at scale. One major advantage of protein design is that we have access to the protein manufacturing processes of nature with the ribosome, like building proteins in the cell. And so whenever we have any protein sequence that we want to make, it's not entirely trivial, but it's much easier to make an arbitrary protein sequence than it is to make an arbitrary small molecule.
That was brilliant. Thank you so much. What an excellent answer. I think we've got Ben back now. Ben, if you're able to turn on your camera, we were really enjoying chatting. Okay, do you want to carry on? Maybe go back. I now can't even remember the question I just asked you. I'm so sorry, but I hope you can, because you were giving us the answer.
Yeah. I mean, the question was like, what is like the future of AlphaFold, right? And I think the best hints you can see is basically on what the preprints and papers they have like started to come out, especially this year. You can see really that they're moving towards not just proteins, but actually like anything that is molecular system, right? Like specifically like, one of the main things why you want to study proteins is like the interaction with small molecules and binders and enzymes. And so like really treating the whole system. And there's David Baker. So I think we can spend more time talking with him. Congrats, I'm a fan of your work.
Thank you so much. David may be listening to the welcome message, but hopefully when he's finished listening to that, we'll get his audio in. But yeah, thank you so much, Ben. I will probably move on now to speak to David. Welcome, David. I hope you can hear us.
Yeah, I can hear you. Can you hear me?
Oh, yes. Fantastic. Thank you so much. We were just having a little chat with all our lovely guests today. And yeah, we've got really into the science. We've done a lot of chatting. I'm going to give you a little introduction. I feel like you don't really need an introduction, but I'm going to do it anyway. David's Professor of Biochemistry and the Director of the Institute for Protein Design at the University of Washington School of Medicine. The Baker Lab develops protein design software and uses it to create molecules that solve challenges in medicine technology and sustainability. Among his recent work is the development of powerful machine learning methods for generating functional proteins. And obviously, David received a Nobel Prize in Chemistry this year for computational protein design. So congratulations. It's absolutely amazing. We're so happy that you've been able to join us today, even if only briefly. We have a lot of people in the audience asking a lot of questions. I'm sure we're going to get a lot more coming in now. But I'm going to begin. We're going to get straight into the science because we are Chemistry World and we've done a lot of introduction already. My first question to you is actually going to be, what are the hardest proteins to design, or actually what makes the properties of a protein easier or harder to design?
Well, let's see. I mean, protein design is really focused on creating new protein functions now, and the difficulty really depends on what you're trying to do. So I would say right now, designing a protein to bind to another protein is pretty straightforward in most cases, whereas other things like designing a protein, designing a molecular machine that will take in a new type of fuel and carry out some new function processively while it's using that molecule is hard. So other things that I think are hard are we're trying to learn how to control mineralization with proteins, because we have all these examples in nature like tooth and bone and shells, but the principles aren't totally understood, so that's hard. So I'd say problems where we're we don't really have complete understanding of the principles are hard. And those are the fun ones too, because we can use protein design to explore those.
Oh, thank you. No, that's a great explanation. And I was going to ask now, I saw your press conference and we were talking a little bit about this earlier with one of your PhD students, Paul Kim, who kindly joined us today as well. You used this phrase in the press conference about the lunatic fringe. And I was saying that this is a relatively new field that's been recognised this year. And obviously you've been working on it for a very long time. But you mentioned in the press conference about being on the lunatic fringe and having to persuade other people that actually what you were doing was going to work, because a lot of people didn't trust that at first. Can you tell us a bit more about that history and that process of getting your research to where it is today?
Yeah, well, I think it's very important in research to always stay at the frontier and, not to, it often seems like you shouldn't work on problems that are too hard because you might fail, but there's also a danger to problems that are too easy because then you don't really learn anything. Yeah, so, Brian Coleman designed Top 7 in 2003, and that was one of the primary things that was cited for the prize. And he really made a big breakthrough in showing that you could design proteins, completely new proteins with new folds, new sequences. And then at that point, the challenge was to design proteins with new functions. And at a certain point, somewhat later, I decided that we were going to try and build everything from scratch rather than take natural protein scaffolds and try and build functions into them. We were going to build absolutely everything from scratch. And that seemed a little crazy because you're taking a big step back. Like, say you want to design an enzyme or a binding protein, you already have some place to start. It seems like it might be better to start with the closest thing in nature rather than start from the beginning. And but it actually turned out that it was good to go back to the start, because then you were kind of building on a solid foundation, and you understood what you were building on. You could control it. But that seemed really pretty crazy because it was a big step back, and but it's kind of an example of and then picking problems which are hard. Like even now, trying to make molecular motors in machines seems kind of crazy, but those are exactly the kind of problems that we should be working on.
Can you actually elaborate a little bit on molecular motors? Because you spoke about that previously as well. And I think it's just a great idea. Can you just give us a little bit more information about what they are?
In nature, we have things like kinesin and myosin that hydrolyze ATP or GTP and use it to power movement. We also have machines in our bodies, like inside cells, like HSP90, HSP70, that are involved in protein quality control. And there are many other machines that do all sorts of different interesting things, and they have in common that there's a fuel that drives alternating conformational changes in the protein that change its interactions with other things. I think that's just a great challenge for protein design now, because, for example, one of the things in terms of therapies, therapies right now are really kind of blunt instruments, like antibodies. They just go and antagonize something. But what if you had something that could hydrolyze a molecule that was in your blood, say after a big meal, and that powered some general cleanup in removal of toxic compounds or disassembling amyloids or other things. So that's an example of something which I would consider at the lunatic fringe right now, which are the kind of problems I like.
That's amazing. I see you were just full of amazing ideas with this stuff. And I'm just wondering, how on earth do you choose which proteins to try and make next? Where do you get your ideas from? Does that come directly from the students that come to you? Do they come with their own ideas, or do you propose things?
I'm really lucky because I get all these amazing, brilliant people from all over the world who come to my group wanting to save the world with some design protein and design a new protein to save the world, and it's also make their fortune. So there are just so many ideas coming in, and then we try and really foster an environment where people are just talking to each other all the time and brainstorming. So new ideas are bubbling up all the time.
That's brilliant. And having spoken to Paul earlier on in your lab, it definitely sounds like you're very involved in the day-to-day running of your lab. But how on earth do you stay across it all? Like, there's so many different projects going on. How on earth can you keep up with it all? It just seems like an incredible amount of work.
Well, I think the answer is I don't really do anything else. I'm there all the time. I read this article a few months ago, think, in the newspaper about how productivity falls after people get Nobel Prizes. And I'm actually working on this email now. I took a screenshot of my calendar where like Monday and Tuesday, like every day I have like it's pretty much meetings with students the whole day. So Monday and Tuesday there was full of meetings, but then Wednesday they were all canceled, and so it was clear problem, real notification problem. But then yesterday, Thursday, and today I'm back to my full complement of student discussion. So I had a fall in productivity, but it didn't last too long.
But for a day or two, okay, we'll forgive you for that. And obviously, it was probably very early on in the day when you found out about your Nobel. How did you receive the news? And I just want to know an insight into that.
Yeah, they actually called my son first. Somehow they found his number, and he likes to sleep with these rain noises on his phone. So he got annoyed because it was interrupted. So he checked his phone, and then he told them to call me. And so actually, my wife picked up the phone, and then it said Sweden, and she got very excited and started screaming. So I couldn't really hear what they were saying, but yeah, it was exciting.
That is absolutely amazing. And I don't know how, you probably haven't even had time to process, what on earth it all means at the moment. And I imagine you are going to be very, very busy, as you already are very busy. But, reflecting on it now and sharing the prize with AlphaFold, I've spoke to, two of the previous scientists we've had on today about the linkages between your work and AlphaFold. And how do you see that progressing moving forwards into the future research you're looking at?
Well, let's see. Yeah, there definitely are some. I'd say that John Jumper's work really inspired us to take a deep, to really start working on deep learning methods for protein design, because he showed that deep learning was so powerful for protein structure prediction. And what really it showed is that the PDB at its current size, with all the work from tens of thousands of people who've put their lives into solving structures, is a sufficiently rich research that you can really learn kind of amazing things from it, and that indeed turned out to be the case, that our deep learning methods for protein design are quite a bit more powerful than our previous sort of physical model based things like Rosetta. And moving forward, I'm looking forward. I talked briefly with Demis and John. I'm looking forward to discuss ways we can collaborate.
That's brilliant news. Okay, I know you haven't got much time, and I'm really, really grateful for you joining us today. One quick question, and it's probably not very quick, but we'll try and do it quickly for you. I know you've got loads of different projects that are being worked on at the moment with your research. What are you most excited about? Can you give us a little bit of a flavour of the kinds of applications that we're applying this kind of science to at the moment, and where it's going to take us, how long we are away from sort of clinical trials or any happening at the moment? A little bit about that.
Oh, well, that's a bit like asking me what my favorite child is.
I was literally thinking that in my head. Sorry.
So, I love all the projects and people, so I'm not going to pick any favorites. I mean, we don't work on things that I'm not excited about. So on the therapeutics, I mean, I'd say broadly speaking, they're projects in medicine, trying to make smarter therapeutics that will not act systemically, but only be active at the time and place in the body when they're needed. And then sort of broad spectrum antivirals for pandemic response. My colleague Neil King is making great progress with vaccines, and we have our first. He has the first de novo designed medicine, which is approved for use in humans, which has gone through all the clinical trials and stuff, which is a COVID vaccine. We have a number of other compounds for cancer that are various stages of the drug development pipeline. We're starting to work a lot on autoimmunity. Outside of medicine, they're really interested in sustainability applications. So problems like, we're making a lot of progress now on catalyst design. So with the latest methods, we're getting new catalysts for new chemical reactions really popping up right and left. It's a super exciting time. And so catalysts for things like plastic degradation of other things in the environment that humans are putting in that we don't want. We're really excited now about approaches to greenhouse gas capture, and then we're doing quite a bit of work now on sort of extending and enhancing photosynthesis and artificial photosynthetic systems. And we're really interested now in, like I gave that example earlier of mineralization. Can we control mineralization, but maybe not just with things like calcium carbonate, calcium phosphate, but with things like zinc oxide, which are semiconductors and make whole new types of materials. We published recently a paper on de novo design nanopores. We're really excited about the sensing applications, incorporating nanopores directly in silicon. So we have totally new types of sensing and sequencing devices. So there's a lot of really exciting applications in medicine and in technology and sustainability.
That's amazing. I mean, I think you've mentioned nearly every problem that we're trying to solve there in the world. It seems, very adaptable.
Well, yeah. What are we missing? I mean,
yeah, I'm wondering that right now. Okay, one final question. It might be a slightly double question. We've heard you don't like to travel much for conferences. Are you going to travel to Stockholm to receive your medal? And the second part of that question is, what are you planning to do with your medal? Because we have an article in Chemistry World that talks about the fate of Nobel Prize medals over the years. What happens to them? What people do with them, and we're interested what you're going to do with your medal.
Oh my gosh, I didn't even realize there was a medal, so I don't know what to do. So I have people ask me how it's going to affect me, and I already had before this an absolute no travel rule. I have to go to one HHMI makes me go to one meeting a year, and that was it. And I will make an exception for this one, but people in the lab are going to have to put up with me every day like they have for the last years, because I'm not going anywhere.
And they'll have to do without you for a few days whilst you go and get that medal and then decide what you want to do with it. You'll have to look at our article to see the fate of some of these medals, whether they get stolen or sold or melted. It's quite interesting what happens. Look, thank you so much, David. We've obviously run over time, but I thought that might happen when you joined us, but that's okay. We've had a brilliant chat with you. Thank you so much. Congratulations. We're really it's really so exciting that you're here. But thank you so much to everyone who's been in the audience watching as well. I'm sure you've enjoyed the chat today. We've chatted to so many different scientists. I'm afraid that is all we've got time for today. But thank you to our guests and obviously David for joining us at the end. Thank you to everyone for being here. If you enjoyed what you've seen today, and then please do check out chemistryworld.com/webinars for all our future content. There's lots of different content on there that you will probably enjoy. Webinars are free to register for and attend, and they're open to all. But from everyone here, we look forward to seeing you in a future Chemistry World webinar. Thank you and goodbye.